Hello dear students, we are done with the glycolysis and Krebs cycle and now we are going to start electron transport chain. In our respiration, we have studied first glycolysis takes place, then formation of histone coenzyme is taking place, and then Krebs cycle occurs into the matrix of the mitochondria. And during these all processes, we are producing ATP and certain coenzymes and electron carriers. They are considered as an NADH and FADH. In order to produce the total amount of ATP, we will have to finally oxidize NADH and FADH so that we can produce total amount of ATP. And in order to oxidize them, there is a chain present in the inner membrane of mitochondria that actually is in a chain of different complexes that receive electron and they are reduce and oxidize, reduce and oxidize. And by this process, electrons will move from one complex to another complex and they will finally reach to an oxygen and they will help in a synthesis of ATP, and that ATP is actually ATP that is formed during electron transport chain. We are also going to happen, what are the complexes and what is the process, we are studying it in detail today. As we have studied, during glycolysis, we have produced NADH. As we have studied, during acetyl coenzyme formation, we have produced NADH. And during Krebs cycle, we have produced NADH and FADH also. Not only substrate phosphorylation is taking place during the process of respiration, but we are also producing these NADH and FADH. All these NADH and FADH are inside matrix of the mitochondria. And from matrix of the mitochondria, they would finally remove electrons. And these electrons will move by this chain that's called electron transport chain. How this process will help and how one NADH and FADH2 are oxidized and how these electrons moving from one complex to another complex is the real story that we are studying today. And I would also ask you to subscribe my channel if you have not subscribed and watch the video till the end so that you can understand the topic properly, and share it with your friends also, and like, share and comment as well, and be with me. Let's learn this most important topic, that's called electron transport chain, because this alone produces a huge amount of ATP from one glucose during the process of respiration. So what actually it is, this is the structure. Let's study first the structure. This is called the outer membrane of mitochondria, and this is the inner membrane of mitochondria. And these enfoldings of the inner membranes are called crusty, and this is a membrane that we made a straight so that we can draw the different complexes and we can study the process of electron transport chain. And this is a space between these two membranes, one membrane, outer membrane and inner membrane. And in inner membrane, this is a space that's said to be inner membrane space, and inside this is a whole region that is said to be the matrix. So this is matrix, this is inner membrane, this is inner membrane space, and this is outer membrane. And in a inner membrane, there are several complexes present. Let's study them one by one what these complexes are. Otherwise, they are named as 4 complexes, but actually there are 6 complexes that are present in this membrane. Well, the first complex that is termed as an NADH dehydrogenase complex. And this NADH dehydrogenase complex is actually a complex that will oxidize NADH and will receive electrons. And this complex is transferring these electrons to another coenzyme that is termed as a coenzyme Q. This complex is not only transferring electrons to coenzyme Q, but it also pumps protons. It is in a hole here that can pump protons from matrix, and it pumps protons inside inner membrane space. So NADH dehydrogenase complex actually is in a place that's called complex 1, which is not only receiving electrons, but it also receives proton, and it pumps electrons in a one way to a cytochrome coenzyme Q, and it pumps proton inside membrane. So this would be the first place where protons have been pumped from matrix to inner membrane space. What happens? This coenzyme has to transfer these electrons forward to cytochrome reductase complex. But we will study another complex here. See, this is a complex 2. And complex 2 is actually FADH2 dehydrogenase complex. It will remove electrons from FADH2. And as these electrons are removed, these electrons are again transferred to cytochrome, to a coenzyme Q complex. Both FADH dehydrogenase and NADH dehydrogenase, they both give, these two complexes give electrons to a coenzyme Q. But actually at this place, FADH dehydrogenase complex can't pump protons. So there is no flow of protons inside membranous space. Therefore we say FADH2 can't pump proton. What actually will happen, electrons either coming from NADH2 or coming from FADH2, they'll come to a coenzyme. What this coenzyme will do, coenzyme can transfer these electrons to another complex that actually is called complex 3. This complex 3 is called cytochrome reductase complex. Because it will receive electrons and it will reduce to this cytochrome C complex, therefore it's termed as a cytochrome reductase complex. When cytochrome reductase complex receives electrons either of the NADH2 or of the FADH2, but it receives electrons from coenzyme Q. When cytochrome reductase complex 3 receives electrons, it not only receives electrons, it receives protons as well, and it pumps protons in one way and electrons to another way. Cytochrome reductase complex can transfer these electrons onward to cytochrome C complex. And when it transfers electrons to a complex C, it also pumps proton from matrix inside inner membrane space. Therefore, this is another place where protons have been pumped inside inner membrane space. These are two places that we are studying where protons have been pumped from matrix inside inner membrane space. What will happen next? Cytochrome C complex is again oxidized by another complex that is called cytochrome oxidase complex. This cytochrome oxidase complex will receive electrons. It will receive electrons from cytochrome C complex. And when this receives electrons, they are oxidized. Right? This cytochrome oxidase complex would finally transfer these electrons to a molecular oxygen that's present inside matrix. It will provide electrons to an oxygen. And when it's pumping electrons in a one way, it will also pump protons in another way. This is the third important place where protons are being pumped inside inner membrane space. But electrons that were moved from NADH or from FADH are finally taken by oxygen. It takes protons from matrix and it forms water. In this way, the excited electrons of NADH and FADH are finally settled with oxygen and water is formed. And during this whole process, actually protons are being pumped. Where from? They are pumped from matrix inside inner membrane space. I repeat again, when NADH is oxidized, it gives electrons to NADH dehydrogenase complex. It takes electrons, pumps them, transfers them towards coenzyme Q, and it pumps protons inside membrane space. And this coenzyme is not only receiving electrons from NADH dehydrogenase complex, but it also receives electrons from FADH2 FADH2 dehydrogenase complex. When this oxidizes FADH2 into FAD+, it is simply giving electrons again to a coenzyme Q. And this coenzyme Q can then give these electrons to another complex that is called cytochrome reductase complex, otherwise complex 3. And when it's receiving electrons, it's just pumping electrons in one way and it pumps protons another way. This is another place where protons have been pumped. Electrons move from cytochrome 3 to cytochrome C complex, but here no pumping of proton is taking place. Cytochrome C transfers these electrons to another complex that is called cytochrome oxidase complex. And cytochrome oxidase complex will receive electrons, will transfer them towards oxygen, and it will take protons and will make water. But when it's receiving electrons, cytochrome oxidase complex not only receives protons, but it pumps, not only receives electrons, but it pumps protons in inner membrane space also. So at 3 places protons are continuously pumped inside inner membrane space, which actually increase the concentration of positive ions, protons inside inner membrane space, which actually triggers another mechanism that's called chemoosmosis. Or this chemoosmosis is actually helping in a synthesis of ATP, which is actually taking place because of oxidation. We term it as an oxidative phosphorylation. With the help of a complex, this complex is called ATP synthetase complex. It is F0N and it has an F1 particle. What will happen from lumen when there is a higher concentration of protons? Protons will enter in this part of ATP complex. And when protons are moving in through this complex, these protons will move from inner membrane space inside the matrix. And this gradient, the concentration gradient of protons is higher in a membrane and it's lower in an inner space, and then we can say protons will move from higher concentration to lower concentration across plasma membrane, across inner membrane of the mitochondria. Therefore, it's in a type of osmosis that is taking place, and it's an osmosis of chemicals. We term it as a chemoosmosis. And the chemoosmosis actually is providing power, it's providing potential, it's providing energy to this complex, and complex stores that energy in a phosphate bond, and that makes ATP combine with P and synthesize ATP. And therefore, we would say all these protons will come in and they will help bind phosphate with ATP and to synthesize ATP, and in this way all the molecules of NADH and FADH formed during glycolysis or during acetyl coenzyme formation or during Krebs cycle, they all will oxidize, they all will give their electrons, and these electrons will move through this electron transport chain, and they would finally move by ATP complex, and ATP complex will synthesize ATP. And this synthesis of ATP is a chemoosmotic oxidative phosphorylation where sufficient amount of energy is produced. If we study, one molecule of NADH is actually pumping 3 protons in an inner membrane space. This one place, 1 proton is pumped. This is another place, another proton is pumped. This is another place, another proton is pumped. It is pumping 3 protons. Therefore, we relate this number of protons with the number of ATP, which is 3 protons are producing 3 molecules of ATP, right? Whereas in case of FADH2, when FADH is oxidized, it gives electrons to coenzyme, but at this stage, protons are not pumped. Just at 2 places, cytochrome reductase complex pumps proton 1 place, and when cytochrome oxidase complex is moving electrons to oxygen, it's pumping protons in. So when FADH2 is oxidized, it pumps 2 protons, and we relate these 2 protons with the 2 molecules of ATP. Therefore, we always say one NADH synthesizes 3 molecules of ATP, and one FADH synthesizes 2 molecules of ATP. This is how all the molecules of NADH and FADH are oxidized during the process of electron transport chain. And this is the story of ETC. Now I'm going to produce the total sum of the energy from one molecule of glucose in my next video. Kindly subscribe my channel, share it with your friends, and let's learn together. Thank you very much.